Hey everybody, PreMedMama here. Thanks so much for tuning in and watching my videos. I'm so glad you're here. For those of you that are new, welcome. If you've already watched my videos, then you know what this is all about. I read aloud through the Kaplan review books, chapter by chapter, section by section. It's a great study aid for learning audio-visually the information that will be tested on the MCAT. Please remember to subscribe to my channel, like my videos, or leave a comment. I appreciate your support. All right, let's get started. Today, we're in Chapter 1 of the Biology Review book by Kaplan, in Section 1-5, learning about viruses and subviral particles. Specifically, I'm going to be talking about HIV, the human immunodeficiency virus. The MCAT wants you to know about this disorder, so listen carefully as we learn and read together. Let's talk first about retroviruses. Retroviruses are enveloped single-stranded RNA viruses in the family Retroviridae. I've mentioned this in my previous video. Usually, the virion contains two identical RNA molecules. These viruses carry an enzyme known as reverse transcriptase, which synthesizes DNA from single-stranded RNA. The process is called reverse transcription. The DNA then integrates into the host cell genome, where it is replicated and transcribed as if it were the host cell's own DNA. This is a clever mechanism because the integration of the genetic material into the host cell genome allows for the cell to be infected indefinitely. And the only way to cure the infection is to kill the infected cell itself. The human immunodeficiency virus, HIV, is a retrovirus that utilizes this life cycle. That's one of the characteristics that makes HIV so difficult to treat. Real-world sidebar on page 27 of your Kaplan review book says, both retroviruses and transduction are under investigation as methods of gene therapy. It is theorized that retroviruses and transduction methods can deliver functional versions of missing or altered genes so that the correct proteins can be synthesized and certain disease states can be alleviated. Gene therapy is discussed in Chapter 6 of our MCAT Biochemistry Review. Next, I'll read through Figure 1-16, which is showing the life cycle of the human immunodeficiency virus. I will provide a link to this image at the bottom of the video. Listen along with me. Efforts to devise vaccines and new treatments for HIV depend on knowledge of the virus's life cycle. HIV invades host cells and commandeers their machinery to make more copies of itself. First, a protein called envelope on the virus must bind to CD4, that's capital C, capital D, number 4, and CCR5, capital C, capital R5, proteins on the cell surface. Number 1. As the virus fuses with the cell, it empties its contents into the cytoplasm. Number 2, a viral enzyme, reverse transcriptase, then copies the virus's RNA genome into double-stranded DNA. Number 3, often making errors that generate diversity in the virus copies. Another viral enzyme called integrase inserts the copy into the host DNA inside the nucleus. Number 4, cell machinery transcribes the viral genes back into RNA, including RNA that can serve as a template for proteins, that travels to the cytoplasm, where ribosomes produce the encoded proteins. That's step number 5. Then, viral RNA and proteins move toward the cell membrane where they gather into budding virus particles. In the immature new virus copy, the HIV protease enzyme modifies viral protein chains enabling the particles, or virions, to mature into a form that is ready to infect a new cell. And there you have it. That is the life cycle of the HIV, human immunodeficiency virus. And last but not least, on page 29 of your Kaplan Biology review book, there's a real-world example related to HIV that the book wants you to know about. It says, HIV must bind to a receptor called CCR5 on white blood cells, and we did just hear about CCR5 in the diagram I read through. It must bind to those in order to infect them. Some people lack this receptor and are thus immune to HIV, which means they can't get HIV. There was recently a case in which an HIV-positive man with leukemia received a bone marrow transplant from a donor that lacked CCR5. Not only was his leukemia cured, but this transplant also resulted in remission of his HIV infection because the white blood cells from his newly acquired bone marrow were not susceptible to HIV infection. How cool is that? Gosh, gotta love science. All right, that's all for now. Thank you for joining me, and be sure to subscribe to PreMedMama.